All right, so that brings us to the final talk of this session. We've got Oscar Rodriguez from the University of Louisville School of Medicine.
Okay, so what we're interested in is in understanding how genetic variation within the immunoglobulin heavy chain locus, or IgH, affects the antibody repertoire. And the genetic variation that we're interested in understanding is genetic variation within coding regions and non-coding regions of IgH of different sizes and types, from single nucleotide variants to small insertions and deletions or indels, to really large structural variants where two individuals could differ by hundreds of thousands of bases. And so the IgH locus is roughly 1 megabase long, contains 130 variable, 27 diversity, and 9 joining genes, an extensive amount of non-functional genes or pseudogenes, and large structural variants that delete or add variable genes into the locus, some of which are highly more frequent in certain populations. So for example, the highlighted structural variant, which introduces 4 genes to the locus, is found at a higher frequency within African populations. And in addition to the genetic diversity that's introduced by structural variants, there's also an extensive amount of genetic variation within coding regions, as shown by the number of alleles per gene. And as we all know, this is really important because during VDJ recombination, alleles from the IgH loci are selected in order to create the antibody. And so in order to understand the relationship between genetic variation within IgH and the antibody repertoire, we sequenced 154 individuals with long-read sequencing, assembled their IgH locus, and from these assemblies, we detected genetic variants. And from the same individuals, we used antibody repertoire sequencing data to calculate clonal gene usage and performed a quantitative trait analysis to identify variants within IgH that are associated with gene usage. And so this analysis lets us correlate the number of alternate alleles with gene usage. And so, for example, in this example, the reference allele is A and the alternate allele is C. And so individuals who have more of the C allele for this genetic variant end up using this VG more frequently in their antibody repertoire. And so across our whole cohort, we identified 8 large structural variants that impact 49% of IgH and end up deleting 41% of the genes. And so these structural variants range in size from 9.6 kb to 284 kb and delete up to 16 genes. So the structural variant in panel B contains the structural variation allele that deletes 284 kb of IgH and 16 genes. The structural variant in panel C has 11 structural variation alleles, and the structural variant in panels D and E all contain novel sequences and genes. And because we assembled the IgH locus, we're able to get these really refined breakpoint resolved structural variants. And so in this example, this single individual has 6 structural variants and 35 genes deleted in at least 1 haplotype. And so on average, individuals have 5.5 large structural variants that delete 257 kb and 26 genes from at least 1 haplotype. And in addition to identifying an extensive amount of structural variants, we also identified a huge amount of SNPs that are missing from SNP databases that are commonly used in the genomics field, such as dbSNP. And so in total, we identified 7,297 genetic variants that were common in our cohort, which we tested against the usage of 80 genes. And so I'm just gonna give a couple of examples. So overall, we found that structural variants had the strongest effect on gene usage. So here I'm showing an example of a complex structural variant where we have 2 structural variation alleles. One structural variation allele has the genes 3-64D and 5-10-1 in one of the alleles, and the other structural variation allele has the genes 1-8 and 3-9. And so individuals who are homozygous for the structural variation allele that contains the genes 3-64D and 5-10-1 end up using these genes in the repertoire much more often than other individuals. and are completely lacking the genes 1-8 and 3-9 from the repertoire. And individuals who have the other SV allele 1-8 and 3-9, the opposite is true. And in this example, we see that individuals who have a haplotype with a deletion in the IGHD region, these individuals use those deleted IGHD genes much less often in their repertoire. Oh, the figure's not showing up, but anyway, it's not just structural variants that have a really strong effect. It's also single nucleotide variants that have a strong effect on gene usage. So here, there's supposed to be like a whole bunch of dots there, but they're not popping up. But anyway, this is a Manhattan plot, the one that was kind of shown earlier today. And what it's supposed to show are SNPs that are associated with gene usage. And on the y-axis, you have the negative log 10 p-value of all these SNV associations. And so the arrow is pointing to a SNP that's invisible, but it's there. And individuals that are homozygous reference for this SNP end up using the gene 3-66 at a 10-fold reduction rate in the repertoire than individuals that are homozygous alternate for this SNP. And this SNP is 777 base pairs away from this gene. In this other example where there are definitely SNPs, we see that the strongest SNP association for the gene usage of B1-2 is 169 kb away, which is kind of pointing to a fact that there could be potentially different regulatory mechanisms that are affecting the gene usage of these genes. And so we were also able to identify genes that were associated with multiple independent variants. And so we selected individuals that were homozygous reference for the SNP that was associated with IGHV 3-66. Oh, there you go. See, told you guys. And then we performed this association all over again, just in these individuals. And we found that there were additional SNPs that were associated with the usage of 3-66 after we controlled for the first association. And we were able to split this group into two groups. One group where we saw complete absence of the usage of 3-66, and another group where some individuals still had some 366 in their repertoire. Okay, and we also identified SNPs that were associated with multiple genes. So there's a SNP in the dotted line there that's associated with the usage of 7 genes. And so in order to understand more about how the gene usage across these genes are interconnected, we created this graph structure where the genes are connected if the same SNP is associated with the gene usage for both genes. And on the right, I'm showing a click of this graph structure that shows that all these genes, their gene usage is all interconnected through a huge set of variants. And this is really interesting given the talk that we just heard. We saw that a lot of the variants that are associated with gene usage, they're actually enriched in transcription factor binding sites for transcription factors that are really important for VDJ recombination, such as CTCF. And additionally, in order to understand if and how variants that are associated with differential gene usage could mediate disease, we overlap the variants that we identified in our data set with variants that have been previously been identified to be associated with disease. And so here I'm just showing 2 examples of variants that are associated with Kawasaki disease and rheumatoid heart disease that are associated with differential gene usage and disease. And here's my acknowledgement, and if you want to learn more, I have a poster 106. Thank you.
Hi, Oscar. You've got a question from the Zoom chat. Is there any plan to follow up some of this data with any kind of epigenetic or chromatin loop studies to elucidate the effects of some of the intergenic variants you found?
Yeah, we definitely want to do that, but one of the critical things that we need is the proper cell types, and the proper cell types here would be the pro-B cells or B cells that are found in the bone marrow. And one of the issues is that in order to do epigenetic studies or epigenetic sequencing, like transcription factor ChIP-seq or histone ChIP-seq, the underlying technology is still based on short read sequencing. So it's going to be hard to scan the epigenome in these like really repetitive regions. We could think about using like the newer long read sequencing technology that also gives you epigenetic information. But yeah, we definitely want to do that.
Really nice talk, very interesting. For the 366, I guess you have no evidence that it's deleted at the genomic level, right?
Yeah, no.
And so you have some of these allelic variants that are affected by this very distant polymorphism.
Yeah.
In the libraries where you looked at expression levels, was that an IgM or IgG library?
Sorry, it's IgM. So we've done all of this in IgM and IgG, and we see that we see the same thing being transferred in IgM, or so we see the same thing in both repertoires. We do see like a smaller depletion in IgG, but we basically see the same thing.
Mm-hmm. Okay, thanks.
And I should say that I've just shown examples here, but we're seeing basically a genetic effect for most of the genes in the Ig loci.
Most of the genes have variants that are
exactly.
Okay, well, that's really important too. Thanks.